everyone, welcome to Biophilia's new chapter video on part 1 of sexual reproduction in flowering plants. So before starting this video, do like, share and subscribe to this amazing channel and do not forget to hit the bell icon so that you do not miss out on any of the future parts. So now let's understand the name of the chapter, that is sexual reproduction in flowering plants. So sexual reproduction to occur in these flowering plants, that is angiosperms, the important part is going to be the flower, right? So let's understand the flower first. तो flower humans के लिए एक बहुत important object है because humans use it in aesthetic, ornamental, social, religious and many other cultural values, right? That was the use for humans, but plants के लिए flowers are going to be morphological and embryological marvels. Now these are called as marvels because flower एक ऐसा organ है that is the site for sexual reproduction in these angiosperms. That is the reason why the heading is flower, a fascinating organ of angiosperms. Now let's see this flower in detail with its parts. So understanding a typical angiosperm, you must know the four whorls or the four floral appendages that are present in the flower. तो ये चार whorls जो होती है, the four layers or the four parts of the flower, they are going to be attached onto the receptacle. Okay, यानी base of the receptacle पे ये सारे whorls are going to be connected. तो चार whorls कौन से होते हैं? First होता है calyx. Okay, calyx is going to be a collective name. और collection किसका है? Of the sepals. Right, these sepals are going to be the whorl which is sterile, and since it is sterile part, it is going to be called as a non-essential whorl. क्योंकि अगर sterile structure है, then it is not going to be a direct benefit for reproduction. That is the reason the calyx is going to be called as a non-essential whorl. Second layer होता है, that is corolla, and corolla is going to be a collection for petals. Now these petals again are going to be sterile structures, and because of the sterility they are going to be called as non-essential whorl. अब last के दो layers आती हैं, these are going to be androecium. This androecium is going to be a collection of stamens, and these stamens represent the male unit of the flower. Now this male unit is going to be fertile, and fertility means it is essential for reproduction. Last whorl होता है, that is gynoecium, और gynoecium में आते हैं सारे carpels. Now these carpels are the female unit of the flower, and being fertile it is going to be a essential whorl. Now in the figure 1 you can locate how the calyx, the corolla, the androecium and the gynoecium actually exist, and this can be further seen in the complete diagram of flower, and you can get the labelings of this flower. Now let's understand the sexual reproduction has three major stages, right? How many stages does sexual reproduction have? Three stages. The first stage is called as the pre-fertilization, right? Second stage होता है the fertilization, and the third stage is called as the post-fertilization. So let's start understanding by the pre-fertilization. Pre-fertilization के अंदर के structures और events we have to study in detail. तो pre-fertilization के लिए the plant needs to develop the required structures for the reproduction, right? तो start कहाँ से होगा? From the reproductive meristem. The reproductive meristem is going to grow to form the inflorescence axis. अब जैसे ही inflorescence axis बनेगा, over that the floral primordia is going to develop. A floral primordia grow होके बनाता है floral buds, and these floral buds when bloom to give us flowers. Now these flowers are going to be an important structure for the fertilization as well as the sexual reproduction. सो फ्लावर में हमें कौन से दो इम्पोर्टेंट वर्ड्स पढ़ने हैं, दैट इज गोइंग टू बी एंड्रोशियम एंड सेकेंड इज गोइंग टू बी द गाइनोशियम. Gynoecium है that is female part of the flower and the androecium is going to be the male part of the flower. So let's start with the detailed study about androecium first. तो एंड्रोशियम के बारे में जब बात हो रही है, so we know androecium is going to have consists of stamen. So it becomes important for us to understand the typical stamen, how does it look and how it is. So a typical stamen is going to consist of two major parts. पहला होता है that is anther and this anther is going to be a terminal bilobed structure. Second part आता है that is filament and this filament is a long and slender stalk, right? और ये filament जो होता है इसका proximal end that is the lower end is going to remain attached to the thalamus or attached to the petal of the flower. Now it depends upon the types of flowers whether the filament is attached to the thalamus or the petal of the flower. Now in over here in the figure 1 you can see a typical anther. Okay, this is a typical anther that is going to be terminal bilobed structure and this slender long structure is going to be your filament and together anther and filament is going to form nothing but that is stamen, right? अब जब हमने पढ़ लिया है द कंप्लीट स्ट्रक्चर अबाउट स्टेमेन, लेट्स गिव अ मोर फोकस टुवर्ड्स एंथर. तो एंथर हमें देखा था दैट इट वाज बाइलोब्ड स्ट्रक्चर एंड व्हाट डज द वर्ड बाइलोब्ड मीन, दैट मीन्स इट इज गोइंग टू हैव टू एंथर lobes. Now these two anther lobes needs to be differentiated and that differentiation can be because of deep groove in the front. Okay, anther के सामने से you can see a deep groove and because of this deep groove the anther is called as bilobed. अब bilobed है, deep groove है, now the two lobes need to be attached to each other, right? So that attachment is going to be provided by the band of vascular sterile tissue that is called as connective, right? Now if the anther is being cut in the transverse section, then what can you see? You can see a structure that is going to be like this, right? And when this type of structure is seen, you call it as four-sided tetragonal structure, right? Four-sided tetragonal structure के corners पे we can see the microsporangia, right? So at the corners you are going to see four located microsporangia, right? Now since the mature anther is going to have four microsporangia, that means anther is going to be also called as tetrasporangiate. So anther के लिए कौन से दो terms use होते हैं? First is going to be anther is a bilobed structure as well as anther can also be called as a tetrasporangiate structure. Now let's see microsporangia. अब जो microsporangia we had made over here that are present at the corners of this transverse section, what is that? On the maturity of the anther, these microsporangia are going to divide itself to form the pollen sacs. और pollen sacs के अंदर क्या होता है? Pollen grains, right? And finally these pollen grains are going to be released out of the anther for fertilization, right? Now let's see about microsporangia in detail. तो microsporangia को पढ़ना is very important because in all microsporangia is the structure that is going to form the pollen grains further, right? So let's see about the structure of microsporangium. Microsporangium is going to be present in the anther and we can see this microsporangium when the anther has been cut in a transverse section, right? Transverse section में when the anther is cut it is going to look like this, right? और इसके अंदर होंगे microsporangia. Now these microsporangia are going to be nearly in circular outline, right? And that can also be seen in this adjacent diagram. Okay, in the figure 1 you can see that the microsporangium are going to be nearly circular. अब microsporangium के आजू बाजू भी कुछ tissues होती है and these tissues are nothing but homogeneous mass of meristematic cells और ये homogeneous mass को special name दिया जाता है that is primary sporogenous cells and this is going to surround all over the anther. अब प्राइमरी स्पोरोजिनस सेल्स जो होते हैं दैट इज सराउंडिंग ऑल ओवर द एंथर दे आर गोइंग टू बी फॉर्मिंग माइक्रोस्पोर मदर सेल्स दैट आर गोइंग टू बी डिप्लॉयड इन प्लॉइडी राइट. डिप्लॉयड सेल्स होने की वजह से दीज विल फर्दर अंडरगो मियोसिस टू फॉर्म द पोलन ग्रेन्स. अब एंथर को जब हमने कट किया था वी आर गोइंग टू गो विद द एंथर वॉल्स. तो एंथर के पास होती है 4 लेयर्स राइट. सो लेट्स स्टडी अबाउट द 4 लेयर्स ऑफ एंथर. एंथर के सबसे आउटर मोस्ट कवरिंग होती है मेड अप ऑफ एपिडर्मिस. Now this epidermis being the outermost is going to be a single layered structure. अब ये outermost है so major function होता है in the protection right. अब यहाँ पे एक fact आता है that is epidermis of this plant is going to develop some fibrous thickenings. Okay, so generally it was having no thickening but particularly in this plant there will be fibrous thickening and then that epidermis since it has been changed it will be called as exothecium right. Second layer आती है that is endothecium. Endothecium is going to be a layer that is going to have alpha cellulosic fibrous bands. Now remember alpha cellulosic है right. You may get confused between the options but to remember these are alpha cellulosic fibrous bands and these bands are going to arise from the inner tangential wall of which अब ये bands why is it necessary because they are going to help in dehiscence. Dehiscence यानी when the pollen grains needs to be released out of the anther तो anther को break होने के लिए these fibrous bands are going to help. Now if some plants that is hydrophytes do not have these fibrous bands that is the reason they are called as hydrophytes do not have fibrous bands. Third layer आता है that is middle layer. This middle layer is going to be consisting of cells which is going to be ephemeral और ephemeral का क्या meaning होता है it is going to last for a very short period of time. Now short period it means when the anther undergoes maturity it is going to degenerate. But middle layer की एक खासियत है that is it is going to be 1 or 3 layered thick right. Last the innermost layer is going to be called as tapetum. Now this tapetum is going to be surrounded by the sporogenous tissue. अब tapetum के जो cells होते हैं उनका major काम होता है to nourish the developing pollen grains. So whenever the sporogenous cells divide and forms the pollen grains the nourishment is going to be provided by the tapetum cells. अब जो tapetum के cells होते हैं they have a very characteristic cell properties that is dense cytoplasm and they will have more than one nucleus. अब जब condition cell के पास ऐसी होती है कि एक ही cell में एक से ज़्यादा nucleus है then we call it polyploidy condition right. अगर एक सेल में एक से ज़्यादा न्यूक्लियस है then there is a high condition that it will have increased DNA content and that can also be seen in the tapetum cells. Tapetum cells के major 3 पढ़ने हैं that is it is going to have dense cytoplasm, it will have more than one nucleus and increased DNA content right. अब टाइप्स ऑफ टैपेटम होते हैं तो टाइप्स ऑफ टैपेटम में पहला टाइप आता है दैट इज सिक्रेटरी और द ग्लैंडुलर टैपेटम. अब जैसे नाम से पता चल रहा है इट इज सिक्रेटरी दैट मीन्स इट इज गोइंग टू रिलीज समथिंग. सो देयर आर 3 मेजर releasing factors. First is going to be sporopollenin. Sporopollenin is going to be the hardest substance present in the plant world. Okay, second release hota hai, that is pollen kit. This pollen kit is a substance that act as glue to stick all the pollen grains together, right? Or third hota hai, that is compatibility proteins, so that the female part of the flower can identify the compatible pollen grains. अब जो सेक्रेटरी टैपेटम के सेल्स होते हैं, दे ऑल्सो प्रोवाइड यूबिश बॉडीज. एंड व्हाट आर दीज यूबिश बॉडीज? दे आर गोइंग टू हेल्प इन द ऑर्नामेंटेशन ऑफ एक्साइन, टू बी स्पेसिफिक, राइट? यानी स्पोरोपोलेनिन जब डिपॉजिट होता है बिकॉज ऑफ दिस टैपेटम, अलॉन्ग विथ इट देयर आर गोइंग टू बी यूबिश बॉडीज. और यूबिश बॉडीज जैसे ही arrange होती है in a specific manner which is going to be special for that particular species of pollen grain, and that because of that Ubish bodies you can segregate the pollen grains basis on their species. Second type of tapetum होता है, that is amoeboid tapetum. Amoeboid tapetum के जो cells होते हैं, they have a property to break down themselves and the entire protoplasm is going to move towards the center. और सेंटर में जैसे ही साइटोप्लाज्म मूव होता है, दैट इज गोइंग टू बी कंज्यूम्ड बाय द माइक्रोस्पोर्स टू गेट नरिशमेंट आउट ऑफ देम. सो मेजरली अमीबॉइड टैपेटम का काम होता है for providing nourishment, whereas the secretory is going to provide extra functionings for the pollen grains. Now let's see a very important process that is called as microsporogenesis. Microsporogenesis क्या होता है? That is going to be formation of haploid spores from diploid microspore mother cell, right? यानी हमने क्या देखा था पहले? Inside the anther there is going to be microspore mother cell which is going to be in its diploid condition, right? Diploid microspore mother cell is undergoing meiosis, right? When the microspore mother cells undergoes meiosis, it is going to form 4 small haploid microspores, right? What are these? These are your microspores. Now microspores production is nothing but microsporogenesis. अब this process कहाँ होती है? This process is going to occur inside the pollen sac. And this complete process of formation of haploid microspores from the diploid microspore mother cell is going to be called as microsporogenesis. अब haploid microspores जो बन गए हैं from a single microspore mother cell, these are going to be forming a 4 colony, right? 4 cells की colony बनाएंगे and this complete structure is going to be called as tetrad. Okay, this tetrad is going to be composed of 4 cells, 4 microspores, and these all are going to be haploid in their ploidy. Now as soon as the anther is getting matured, और जैसे ही maturity आती है anther में, it starts to lose water, right? So it gets dehydrated, and because of dehydration what will happen? It is going to burst open. Now when the anther burst open, क्या होगा? तब तक pollen grains develop हो जाते हैं from the microspores. So these were the microspores in their tetrad condition, right? Tetrad condition से when they undergo development, what will happen? They will form the pollen grains. Now undergoing this process क्या होता है? Anther mature होगा, and when the pollen grains are completely formed, the anther is going to dehisce, and that would lead to spreading of thousands of pollen grains at a time, right? So this complete process is going to be called as microsporogenesis. Now let's understand a very important part, that is pollen grain. अब pollen grain क्या होता है? Pollen grain is a structure that is going to represent the male gametophyte. Okay, याद करो pollen grain कैसे बना था? That was made from a microspore mother cell, right? Microspore mother cell से बना था after meiosis, right? Meiosis के बाद बना था, and that led to formation of tetrad of microspores. Microspores बने थे in the haploid condition, whereas microspore mother cell was diploid condition. अब जब पोलन ग्रेन्स बनते हैं, what is going to happen? They are going to carry the half amount of DNA of the plant body. That means pollen grains जो होते हैं, they are going to be the representative of entire male plant, right? Complete genetic information is going to be comprised in this one small pollen grain. That is the reason it is going to be called as male gametophyte. अब पोलन ग्रेन्स जनरली दे आर गोइंग टू बी स्पेरिकल इन स्ट्रक्चर और उनका साइज होता है अबाउट 25 to 50 माइक्रोमीटर इन देयर डायमीटर. सो इट इज अ वेरी स्मॉल स्ट्रक्चर एंड इट इज वेरी डिफिकल्ट to see its complete structure with naked eyes. So microscope के अंदर if you get a chance to see it under microscope, it is going to look very fabulous because of various designs that it forms because of the ubish bodies that were released by the secretory tapetum, right? So different pollen grains look differently. So you can see in the adjacent figure over here that all the pollen grains are going to look different, और ये different होते हैं because of the ubish bodies. अब pollen grain का जो cell wall होता है that is going to be called as the sporoderm, और जो sporoderm होता है उसके majorly 2 layers होती है, right? How many layers? 2 layers. So you can divide sporoderm into 2 layers. पहली layer होती है that is called as exine. Exine E से याद रख सकते हो, exit the outer layer, right? So exine making up the outer layer. तो outer layer जो होती है that is going to be made up of sporopollenin, और sporopollenin कौन रिलीज कर रहा था? That was the secretory tapetum. स्पोरोपोलिनिन का एक वेरी स्पेशल कैरेक्टरिस्टिक याद रखना है, दैट इज इट इज गोइंग टू बी मोस्ट रेजिस्टेंट ऑर्गेनिक मटेरियल्स नोन टू ह्यूमन बींग्स, राइट? टू रिमेंबर यहाँ पे ऑर्गेनिक याद रखना है बिकॉज इन ऑप्शंस यू कैन गेट ऑप्शन वेर इन इनऑर्गेनिक कैन ऑल्सो बी रिटन. सो इट इज important to remember organic word over here, right? So that was a little tip to remember while solving MCQs. आगे बढ़ते हैं with exine. So exine is made up of a material which is very resistant, and that resistance is given for high temperature, strong acids and alkali. In these conditions also sporopollenin की वजह से the pollen grain can survive. Until now there is no enzyme known to humans that can degrade sporopollenin. Because of so many characteristics of this exine, it becomes easier for the pollen grain to have a better chance of fertilization. अब exine जो होता है उसके कुछ advantages या characteristics बोल सकते हो that you need to see in this table. तो यहाँ पे क्या याद रखना है? एक्साइन जो लेयर होता है दैट इज गोइंग टू हेल्प इन फॉसिलाइजेशन. फॉसिलाइजेशन में कैसे हेल्प करता है? बिकॉज ऑफ द स्पोरोपोलिनिन द कंप्लीट पोलन ग्रेन्स आर वेल प्रिजर्व्ड. अंदर का मटेरियल जो पोलन ग्रेन के अंदर का जेनेटिक मैटर है दैट इज नॉट गोइंग टू बी डिग्रेडेड बिकॉज ऑफ दिस स्पोरोपोलिनिन, and because of that you can even study the thousands of years old pollen grains even now, right? अब स्पोरोपोलिनिन या एक्साइन की वजह से the hard covering of the pollen grain is also going to protect it from the hazardous environment. Suppose pollen grains release हो गए and the outer environmental conditions not get favorable, but still because of this hard covering it has a chance to survive. अब थर्ड important point याद रखना है that is it is going to exhibit a fascinating array of patterns. जैसे you can see in the adjacent figure that there are going to be various patterns and designs, and because of these designs to be very significant for the specific species they are going to be a taxonomic significance. That means every species is going to have different type of pattern, और ये different patterns की वजह से people can study them and understand from where which species the pollen grains are. So that makes it a taxonomic significant. Fourth point आता है, that is enzyme जो है that is not present all over the pollen grain. There are going to be some places wherein sporopollenin is going to be absent, और जहाँ पे the sporopollenin is absent हम उसको बोलते हैं germ pores. Now a very special point to remember about germ pores is, अगर हम बात कर रहे हैं germ pores की, and we know that there are going to be two various plants, right? So first we have the monocots and we have dicots, right? तो monocots के केस में there are going to be one germ pore, यानी एक ही ऐसी जगह होगी wherein there is going to be lack of sporopollenin, and because of one germ pore you also call it uniporate. What do you call it? Uniporate. Now in the case of dicots, there are going to be three germ pores, right? That means three places wherein sporopollenin is absent. अब three है तो you call it triporate, right? I hope you can remember this point regarding germ pores. Now let's move on with the second layer of the sporoderm, that was intine. Intine is going to be the inner wall, and this is going to be a thin and continuous wall, और ये जो intine होता है that is going to be made up of cellulose and pectin. Again this cellulose and pectin is going to be one of your questions, right? So you have to remember this. अब इंटाइन जो होती है that is made up of cellulose and pectin, जबकि एक्साइन जो होती है वो किससे बनेगी, that is going to be made up of sporopollenin. अब जैसे ही ये दो layers finish हो जाती है, what is going to come next? That is going to be your cytoplasm, and this cytoplasm is going to be surrounded by the plasma membrane, right? So this completes our study about pollen grains. Now let's see some related topics about pollen grains. तो पहला टॉपिक डिस्कशन में आता है that is pollen allergy. अब pollen allergy से अगर आप terms separate out करो, so you can understand that because of pollen grains some people can have allergies, right? So pollen grains of many species of plants, generally anemophilous plants. Anemophilous plants किन्हें बोलते हैं? Plants that have air pollination. Okay, plants undergoing air pollination are going to be called as anemophilous plants. Now because of air pollination, plant is going to release pollen grains into the atmosphere, और जैसे ही ये plant के pollen atmosphere में आते हैं, and if the person inhales, then the person can have high chances of several allergies, bronchial infections, and in severe cases the person can also develop chronic respiratory disorders like asthma and bronchitis. So people who are more prone to get allergic, they need to stay away from these anemophilous plants. तो यहाँ पे 3 एग्जाम्पल्स है, those can cause pollen allergy. पहला होता है पार्थेनियम, that can also be called as carrot grass. सेकेंड होता है amaranthus, और थर्ड है chenopodium. These are the plants which are going to cause pollen allergy. Now a very important point regarding parthenium is going to be, parthenium जो है that was not originally found in India. वो गलती से आ गया था in India as a contaminant, यानी it was not purposely brought, but by mistake along with the imported wheat it had been introduced to India. Now because of this parthenium, many people suffer from these pollen allergies. सेकेंड टॉपिक जो हमें पोलन ग्रेन्स के रिलेटेड पढ़ना है, दैट इज पोलन प्रोडक्ट्स. सो पोलन प्रोडक्ट्स, why do people use them? Because people think that pollen grains are going to be rich in nutrients, right? Because of their rich in nutrients, they claim to increase performance of athletics and race horses, and because of this property it has become a fashion to sell pollen grains as in the form of pollen tablets, और ये pollen tablets people consume करते हैं as food supplements, right? So Western countries may many people have a habit of consuming pollen grains in form of tablets or syrups or dried pollen grains, which are easily available in the market. थर्ड यहाँ पे हमें पढ़ना है दैट इज पोलन वायबिलिटी. पोलन वायबिलिटी इज अ टर्म दैट इज डिफाइंड एज period for which the pollen grains retain the ability to germinate on the landing of stigma, and that is called as pollen viability. अब ये तो हो गया इंग्लिश का स्टेटमेंट, बट what does it actually mean? That means from the time that it has been set out from the anther till the time it reaches the stigma. ओके यानी जब एंथर में से पोलन ग्रेन एटमॉस्फेयर में आया एंड टिल द टाइम इट रीचेस द स्टिग्मा, ओके, एंड इट हैज अ एबिलिटी टू जर्मिनेट, यानी फर्टिलिटी की एबिलिटी है उसमें, दैट पर्टिकुलर टाइम इज गोइंग टू बी कॉल्ड एज पोलन वायबिलिटी. सो पोलन वायबल कब है कब नहीं है, दैट ऑल डिपेंड्स अपॉन द टेम्परेचर एंड द अमाउंट ऑफ ह्यूमिडिटी दैट इज प्रेजेंट अराउंड द plant. Pollen viability differs from plant to plant, but generally wheat and cereal, okay, wheat and rice that comes under the cereals category, those pollen grains have the viability of about 30 minutes, that is half an hour. Yani from the time it is released from the anther, वहां से 30 मिनट्स काउंट करने तक अगर वो नहीं पहुंचा टिल द स्टिग्मा, देन इट लूजेस इट्स एबिलिटी टू जर्मिनेट एंड फर्टिलाइज द फीमेल पार्ट. जबकि families like लेग्यूमिनोसी और रोजेसी और सोलानेसी फैमिली, these families के जो members है, they have their pollen viability about several months. Now people need, those who are undergoing artificial hybridization, they have a test that is called as quick test of germination for checking the pollen viability. तो viability of seeds test करने के लिए what is the technique used, that it is released into a solution that is 2,3,5 triphenyl tetrazolium chloride. When this is used, then the person can identify whether the pollen or the seed is viable or not. फोर्थ कॉन्सेप्ट आता है अबाउट पोलन बैंक्स. पोलन बैंक्स एक ऐसी जगह है वेर इन यू हैव द सोर्स फॉर स्टोरेज ऑफ पोलन ग्रेन्स, राइट? These pollen grains, because of their sporopollenin, they can be stored into liquid nitrogen, और liquid nitrogen का temperature याद रखना है, that is going to be -196 degree Celsius, right? So because of such a freezing temperature, these pollen grains can be stored. और ये स्टोर करने का पर्पस क्या है? पर्पस इज सो दैट इट कैन बी यूज्ड इन प्लांट ब्रीडिंग प्रोग्राम्स later on. यानी pollen grains अभी है, you store it into this deep cold freezing liquid nitrogen, and then you can use it later on when you need to fertilize the female plant. This complete process of freezing the pollen grains is called as cryopreservation, right? And this cryopreservation technique is majorly used in pollen banks to store the pollen grains. Now let's understand the structure and the development of the male gametophyte. So starting with the sporogenous tissue. Sporogenous tissue, when is going to differentiate itself, it is going to form the microsporangia. अब microsporangia के अंदर होते हैं microspore mother cells. Now these microspore mother cells are going to be diploid in their condition. Now these diploid microspore mother cells, when undergo meiosis, they are going to produce haploid microspores. Now these haploid microspores, when are going to get covered with sporopollenin, they are going to form the pollen grains. Now these pollen grains can either be shed or released from the anther in the 2-celled stage or the 3-celled stage. Generally 2-celled stage condition is seen in dicots and 3-cell stage condition is seen in monocots. Now this all depends on the lifespan of the plant, क्योंकि dicots में lifespan ज़्यादा होती है, so the pollen viability automatically increases, so the pollen gets some chance to get divided as soon as it reaches the stigma. जबकि मोनोकॉट्स के केस में क्या होता है, लाइफ स्पैन कम होने की वजह से ऑटोमेटिकली पोलन वायबिलिटी डिक्रीजेस, pollen viability decrease होती है, so the plant thinks that we will release the pollen in a mature condition कि जैसे ही वो स्टिग्मा पे पहुंचे the fertilization occurs and the new offsprings are formed. That is the reason monocots are going to release their pollen grains in 3-celled stage, whereas dicots prefer to release it in 2-celled state. तो अब ये 2-cell stage जो होती है, what are these 2 cells? These 2 cells are going to be formed because of mitosis in the microspore. तो माइक्रोस्पोर के अंदर जब माइटोसिस होता है, that is going to produce the vegetative cell and the generative cell. अब जो वेजिटेटिव सेल होता है, that is going to be bigger in size and having abundant food reserves, because this is the cell that is going to provide energy as soon as the pollen grain releases from the anther. Now this generative cell, vegetative cell, is going to have a large irregularly shaped nucleus. अब जनरेटिव सेल की अगर बात हो तो जनरेटिव सेल is going to be smaller in size and a spindle shaped cell that is going to float in the cytoplasm of the vegetative cell. अब जनरेटिव सेल के पास एक न्यूक्लियस होता है and will have a dense cytoplasm. So this was the 2 cells that were present in the 2-celled stage of the pollen grain. So generally 2-celled stage जो होती है, that is seen in 60% of the angiosperms, whereas the rest of the 40% are going to have their generative cell dividing to form the 2 male gametes. अब 2 male gametes और एक vegetative cell मिला के बनाते हैं the 3-cell stage. तो अब जो 2 male gametes की बात हो रही है, how are these male gametes? These male gametes are going to be non-motile and amoeboid in shape. Now because of the division, slightly they can be unequal in size, but because of the 3 cells, that is the pollen having 2 male gametes, that will be called as a 3-celled pollen, or it is called as the mature male gametophyte. Correct? Now let's understand it in the diagram also. The diagram में we had understood about exine the outer covering, the intine the inner covering, फिर न्यूक्लियस था और जर्मपोर, that was the absence of sporopollen in area. फिर the microspore is going to start dividing its nucleus and form 2 cells, that is going to be the vegetative and the generative cell. फिर अगर generative cell के अंदर अगर division हो जाए forming 2 male gametes, then that is going to be your 3-celled condition. Okay, so I hope with this explanation all the properties of the androecium, that was the male part, gets completed over here. So in this video we did complete about the androecium. I'll come back with one more amazing session in which we would cover the gynoecium part of the flower. Till then you have to like, share and subscribe, and do not forget to hit the bell icon so that you can study the gynoecium with biophilia. Stay happy, stay smiling, keep studying till the next video comes. Thank you.